So we're going to talk a little bit about the polymerization chain reaction, which is commonly known as PCR. It's a very common technique in microbiology, and most of what we're able to do with DNA would not be possible without it. So to start with, what is it? It's basically using the DNA replication machinery of a cell to make multiple copies of a specific DNA sequence, but it doesn't have to be done inside a cell. It can be done inside a little test tube. It's probably the most successful technique in biology, lab technique. You can take a trace amount of DNA that might be found at a crime scene, for example, and you can make as many copies as you want for testing. In the old days, before PCR, most of the DNA tests that are now done were never done on crime scene evidence, for example, or for paternity testing, because the tests would destroy the sample. In order to test DNA or sequence it, you have to destroy the piece of DNA. Well, if you only have a limited amount, you don't want to be destroying that little bit of a sample, because if you do it wrong, for example, you might get the wrong result and then you're out of evidence. So until PCR came along, DNA evidence wasn't really used in courts, for example, or in crime scene investigation, or in paternity testing, or any of these things, because they didn't want to destroy the samples. Well, then we have PCR, and now we can do all of those things because we can make as many copies of a target DNA sequence as we want. It's used anytime you need to make large amounts of DNA for analysis, but you only have a small amount to start with. So we can use it for paternity testing, for example, to establish blood relationships, to identify remains, to help convict criminals or exonerate the falsely accused. It's an essential procedure. You almost can't do any other genetic test without first running a PCR experiment to increase the amount of DNA that you want to test. So this technique is not as old as you might think. It was actually discovered in the early 80s. Scientist, chemist, who happened to be working in a genetics or microbiology lab named Kary Mullis in California happened upon this particular technique. He published a paper in 1985 describing it, and since he was working for the Cetus Corporation at the time, they technically owned the rights to PCR. They were able to sell it to the general public for $300 million. Mullis ended up getting a $100,000 bonus, but he did win the Nobel Prize in Chemistry in 1993 for his discovery, and since then he's become sort of a minor celebrity in the science world. He's a very interesting guy, Kary Mullis. If you want to read a good book, I would recommend a story. It's sort of an autobiographical sketch of his life called Dancing Naked in the Minefield, and it's very interesting. He's a very strange guy, but brilliant. So if you want to do PCR, here's what you need. You have to know the parts of the target DNA sequence that you want to copy, that you want to amplify. And the reason you have to know what parts of the DNA sequence you want to amplify is because you're going to need some synthetic primers. Remember that DNA replication requires primers in order to replicate the DNA. Well, we're going to make primers in a lab, synthetic primers. And if you want to make a primer that will fit in there, you have to know parts of the sequence that the primer can be attracted to. So we're going to need two types of synthetic primers, complementary to the two ends of the target sequence. The one end of the lagging strand and the one end of the leading strand, so that you can get your DNA copied. You're going to need lots and lots of the DNA nucleotides because you're going to make multiple copies of this stuff. So you're going to need a lot of copies of that DNA nucleotide, the DNA nucleotides. And finally, you're going to need something called TAC1. The biggest stumbling block when they first figured out PCR and how it was going to work was the fact that it has to be done, portions of the PCR experiment have to be done at high temperatures. And high temperatures are really, really bad for proteins. Proteins don't do what they need to do at high temperatures. They unravel, they stop working. This is why a fever, high enough fever, can be deadly, because all of your proteins, those enzymes, stop working. So what they needed was heat-resistant DNA polymerase. Human DNA polymerase wasn't going to work because it's not heat resistant. It's very sensitive to temperature changes. So they went looking for an organism that could live at very high temperatures, and they found something called Thermocycler, which is a bacteria that lives near volcanic vents where the temperatures reach almost boiling. And the bacteria, Thermocycler, lives perfectly naturally at those temperatures. So that means that all of its normal proteins and enzymes have to function at high temperature. So they just take the DNA polymerase enzyme from that organism. And then when they have to heat up the PCR run, it's no big deal. DNA polymerase will still work at a high temperature. It's done at high temperatures for part of the run, and all of this happens inside an instrument called the Thermocycler. A thermocycler is an automated computer-run instrument, and all it does, if you think of the word thermocycle, all it does is it varies the temperature. It cycles through different temperatures, and different temperatures have different effects on DNA. So we'll take a look at a little animation to show you how this all works. So once you have all of your items, you first are going to raise the temperature. And this causes the DNA to split apart. That's called denaturation. Once the DNA is split apart, you lower the temperature a little bit, and that allows the primers to come in and stick to the ends of the gene that you want to copy. That's called annealing. Then we bring in the Taq polymerase, and the polymerase creates the new strands of DNA by using up these copies of the DNA nucleotides. We cycle up the temperature again. It causes the two new molecules to separate. We cool it down. We allow the new primers to come in again. In comes Taq polymerase and creates another copy of each of those strands. And now we have 4 copies. We raise the temperature again, we split them, and we repeat the process. Every cycle of change in temperature doubles the number of DNA pieces that you have of this particular gene. A cycle takes about 20 minutes. So you run a few cycles and you'll have plenty of DNA.